LuxR are bacterial receptors that bind specific small molecules or ligands to induce a behavioral change in triggering environments. This response manifests as change in the transcription of genes. We can leverage this mechanism to trigger a targeted response with synthetically designed ligands. Let's analyze one design. Start a new graph to design a pipeline analyzing LuxR binding. Upload the genetic sequence encoding the LuxR receptor. Start by selecting the Gene FASTA node. Define which uploaded genetic sequence to work with. Translate the DNA to a protein sequence in order to simulate the receptor. Set the codon type to bacterial for a species-specific translation. To search for known protein domains of interest, add the interproscan annotation. A sequential viewer will provide visual insight into found domains of interest. Predict and simulate the 3D structure of the LuxR receptor using the OmegaFold algorithm. Now let's integrate a branch to analyze the binding to a naturally occurring ligand. Select the SMILES to SDF node, converting a string containing chemical structure information. A SMILES string of a known LuxR ligand is easily found on PubChem. Add a docking node to predict the protein ligand binding. Input the protein structure prediction from OmegaFold and the ligand structure. To visualize the protein-ligand complex, the structural data has to be merged into one. For that, merge the protein structure and the ligand docking predictions. Select the Protein Data Bank viewer to visualize the predicted 3D protein-ligand complex. Add a protein-ligand binding affinity calculation for an initial quantitative binding assessment. Select the corresponding viewer to directly see results. Now create the same analysis branch for a synthetically designed ligand. Start with the chemical structure of a synthetic ligand translated into a SMILES string. Now reconstruct the same protein-ligand binding analysis pipeline branch. You can control individual edges connecting the tools in the pipeline. And then execute the graph. After computation is complete, view the results directly in the notes. Let's check for found annotated protein domains in the target protein LuxR. Integrated redirection to the online database provides further information about the found domains. Now let's look into the LuxR binding to its naturally occurring ligand. You can choose between various visualization styles. Integrated visualization plugins enable direct analysis without the need for additional applications. Inspect the high-resolution structural representation of the bound natural ligand. Binding affinity calculations show comparable metrics for protein interaction of the two ligands. Let's take a look into how the synthetically designed ligand bound the receptor. 3D visualization shows that this ligand may interfere with the protein structure. Let's integrate these computational results into the next ligand design iteration.